With precision health or personalized medicine, also they call it, it's like having a unique treatment plan for every patient, right? And kind of goes back to this seminal way of doing blood transfer between patients. And imagine if we didn't know the blood type, and how could we do a blood transfusion? So that was a big step in medicine, and I think precision health, where we can take individual variability in the genes and environment and lifestyle, would be equally phenomenal as we make progress in research and make that work. So one size does not fit all. We all know that, and but how to make it more customized beyond that description is what the challenge is. So this concept is known, but to implement it in practice is really difficult. So let's see what we as computer scientists can do for this lofty goal. The way I think about it is it starts at the data sources. So you have heterogeneity and diversity in the data sources. So there's electronic health records, which is today existing, right? So they're already used today for making a lot of decisions. But then there's your genome data, epigenomes, like how these genomes actually manifesting, microbiome data, which is a lot of we have trillions of microbiomes in our body, and they really shape how the genes manifest and how the chemical and biological processes in our body manifest. And metabolomes, which are small molecules like lipids and amino acids, and they also make a huge difference to how our body reacts to different bugs and germs, and also how we kind of age and so on. So these are the various data sources, and then there should be some way of converting all this. Most of it is analog, actually, like the genomic data, biological. There should be some way to digitize it, right? That's when we can actually build a system around it. So there has to be digitization process, which is where sequencing comes in. Sequencing is a way to digitize genome and even epigenomic data like RNA. And then we have decision support tools which use this digitized information and then do analysis on it. And this lot of data which is generated in this, if we try to collect all this cumulative data, just to give a perspective, Facebook probably collects a gigabyte per user, but then a human genome is 300 gigabytes, and if you do liquid biopsy, that's a terabyte of data. So the scale of data is very huge here. So how do we make meaningful analytics on it is a computational problem as well. And then once you do the analysis, then you can do diagnosis, you can do prevention, you can do treatment and management. So one thing I would like to emphasize here is it's not just only in a clinical setting as a doctor's office where these platforms are useful, but they can be offline too, at the customer, for a user, for a specific user, and do personalized decisions outside the hospital. So that's a precision health platform. And if we think about different problems here, one thing which I'm excited about is efficiency, and I'm going to talk about that. But outside efficiency, security and privacy is also a huge problem. We are sharing very personal information, and how do we share that with like the precision health platforms is an important problem. And form factor, we want to make these small so that they can be at the point of care, at the user, for consumer products. So form factor is also very important. So I think these three problems are very interesting as well as very important to solve for precision health platforms. So as I was talking before, I'm going to focus on efficiency, and one of the things which I'll focus on is digitizing these analog signals using sequencing. So sequencing is a very key ingredient here of precision health, right? That is at the interface of biological data and digital data, which we need to analyze. And also it's a very important form of signal which we can provide for precision health. So let's take a look at how sequencing has evolved and so on. And I'm sure there have been a lot of talks today morning going over this stuff. So I'll just build a little context and rush through these. But we have come a long way since the first genome was sequenced, and actually 1,000 genomes were sequenced not too far away, probably 15 years back, right? But it's growing exponentially since then. We have moved on from second generation to third generation sequencing, which is, even like has reduced form factor. And going forward, this cell sequencing is going to be even a more cooler technology, because now you can actually target individual cells from different parts of your tissue or organ systems and make meaningful specific diagnosis of that specific organ system or that specific organ, right? So I think that's a very powerful technology, and good and bad, it's computationally very intensive. So that's also there, and there's a huge momentum towards sequencing every citizen in many countries, right? So that there's a lot of genome sequencing which is going to happen in the next 5 or 10 years in population scale. So what is interesting is that as the cost of human genome is falling, because we are going on this exponential trajectory of sequencing more and more genomes, and it's $1,000. I believe it can fall to $100 too if we are, it depends on what application we are looking exactly for, like how much sequencing we need to do. But silicon is not scaling that well, right? So there's a gap between the cost per human and how fast our silicon is scaling. So we need to bridge that gap, and that's where hardware, software, co-design, and acceleration would help. And there's exploding sequencing applications. So we and every domain will need a very custom computation pipeline, hardware software pipeline. So for instance, we just went through the pandemic, and there we have to sequence viruses and do epidemiology analysis, and kind of it's a more of a classification problem along with sequencing, right? This is where we did our work on squiggles filter. Then there's consumer genotyping, that's kind of a more lightweight whole genome sequencing. There's cancer treatment where we want to diagnose and figure out these mutations. Microbiome, sequencing your gut microbiome or microbiome all over your body. Again, that's a different problem from cancer treatment and genotyping. And then, agricultural sequencing, it was a surprise to me when I first found out that the wheat genome is like much, much larger than the human genome. So, that's another, like there's many special custom sort of techniques we can come up there again, algorithmic and hardware software for a different, species and different domains. And then one thing which I'm also excited about is real-time sequencing. So in operating room sequencing, which we have done some work, and I'm going to talk about that today. So I won't go so much here. And then food safety. Sometimes, there was the story I tell, like Chipotle found some bugs in the meat that they were transporting, and they had to throw off millions worth of food and fresh items. So, kind of doing that kind of sequencing during that supply chain pipeline is also very important for food safety to save our losses. And this is the best way to, this is one of the most accurate way of diagnosing any problems with food safety. And liquid biopsy, this is ongoing work in my group, so we have not yet published it, but I'm very excited about that because it's, again, a needle in a haystack and a very different problem from all the other sequencing applications which we have seen here. Like portable pathogen detection is more a point of care version of epidemiology analysis, or figuring out new pathogens quickly or even existing pathogens more quickly, like kind of before PCR primers can be developed and deployed all over the world. If you want to still if a new pandemic is emerging, how do you have these portable pathogen detectors? So with that, I'm gonna do a little deep dive into whole genome sequencing. This is where we started out in our group, and I'm gonna talk a little bit about the work we have done here, and then I will talk about the in operating room sequencing. So probably I'm preaching to the choir here, but still I'll repeat the steps here just for the context. You get a sample, right? It could be a tissue or a plasma or a liquid sample, and then you have to do some sample preparation, and then you sequence it through a sequencer. And the sequencer, basically what it does is it breaks down your very long genome into many small fragments, and it duplicates it many times over. And the reason we are duplicating it is for error correction. Although the error rates are very low with short read technology, you still need to do that to kind of tease out the errors versus the actual variants. And then, since we have a lot of these short fragments, we have to find a reasonable location for them on the reference genome, and that process is the read alignment. And read alignment has got two steps which I'll talk about soon. And then once you kind of find an approximate location for these reads, then we kind of remove all the duplicates. We do some sorting to make sure they're in a reasonable order and format for the variant caller to call the variants. And then what the variant caller essentially does is it piles up all these aligned reads together and then finds out true variants. That's the whole goal of this exercise, is that if you have a reference genome, you want to see where in your genome the base pair is different, right? In this case, CNA. And make sure that that is systematic, systematic difference, and the random differences are basically pruned out because they are just from the errors. And using this variant information, then we can do diagnosis. So what I'm going to talk about is the read alignment step today. We kind of looked at a very, this is maybe 1 or 2 years old, but it takes about 14 hours on a very high-end CPU with lots of cores, 32 cores, to do all this thing, the process. So we want to kind of reduce the time and also the energy to do that. And then when we want to do this in parallel for lots of genomes, we want to increase the throughput as well. So we want to reduce both the time to result as well as throughput. So the first work I'll talk today about is GenX. So this focuses on, once you have figured out the location on the genome where this read would approximately align to, we do an approximate string matching step. And what is state of art today is some variation of Bandit-Smith-Waterman. That's what most software implement, with different scoring functions. And what we looked at, and we came from a very different background. We're trying to look at the Micron's Automata processor initially and trying to see if that can solve this problem. And we quickly realized that's not possible. But we wanted to build an automata to kind of solve this problem. And the couple of key insights which we had was that we wanted, if you build an automata for the entire human genome, you have to encode all these substrings in genome, and slowly the automata state just explodes because there's so many different strings once you break it down. So we wanted to make a string independent automata, and it has to have local sort of scoring. It has to have a local connections so that the automata is efficient. So that's what was a key insight which led to Scylla. Of course, to make it work, I think, to find out the state transitions and the differences between the input query and the reference, we had to create some retro comparison codes which would make this very nice and local, and the interface, the query and the reference interface, to be very clean. So that's how we ended up with GenX. And at that point, we also wanted to just not do the string matching, but we wanted to actually figure out the optimal path. Only finding the best score was not sufficient. We have to find out the traceback path. So then we kind of augmented this systolic array so that it can do traceback. And also, supporting affine gap scoring was not easy. It really bloated up our little tiny piece. So, the one which we had for edit distance was beautiful and small, but once we had to put in all these features, then it started becoming a little bit larger, but then that's realistic. That's what we really needed for the high accuracy and bit exact results. And we not only built the architecture for it, we also fabricated a chip for this and showed its utility. Although it was in 55 nanometer, it was still so much more efficient than the CPUs and GPUs, is what we measured. So that was the GenX sort of architecture. So after building GenX, what we realized was this order of k-square can be very innovative in terms of area. So that sort of motivated our next work, which was CDEX, which we went with like a little bit different design, but the whole idea was now we'll go with sort of a banded implementation with just a PE which can process the wavefront of the wavefront is shown here, of the wavefront of the optimization dynamic programming problem we're trying to solve with the alignment. But what we realized was that, when you're doing this so nobody does full band, right? So even in the software, they try to find the fixed band, which depends on the query string sizes. So full band is not, Genax supported full band, but we quickly realized that's not efficient. So the band in software is usually measured based on the query string size. So that's also not very optimistic. But if you look at all these different paths which go through for to find the optimal path, a narrow band is mostly always sufficient. And narrow band reduces, the number of P cycles, the number of Ps needed, and so on. So it's more efficient. However, there is a slight chance that when you go with a narrow band, you'll miss the optimal path. So, as architects love speculation, what we came up with is that, we wanted to get the optimal path, but at the same time, we want to make sure we catch these sort of small, deviations at the runtime. So we came up with a speculative method where we would execute only the narrow path optimistically, hoping that it will catch all the paths. But we computed, and this is a DP formulation with a fine gap scoring, so it's possible to compute the thresholds outside the band. So if the score at any time inside the band is exceeding these threshold scores, means that your path has deviated and has come out of this band which we are computing, the narrow band. So having those threshold scores gave us a very cheap way of doing the checks whether our speculation is going to be successful or not. And that worked very well in this region, which I've shaded. But if your reference string is typically much larger than the query string, so this is bottom half of the DP matrix, which was very hard to compute a threshold for. So there, what we said is, okay, let's give up with the threshold scoring of the fine gap, and we created an even more optimistic edit distance space score, which would help us catch that. And for that, we had to build a very tiny edit machines, which was smaller in size than the fine gap scoring. So their static threshold scores was not sufficient, so a tiny little edit machine checker would compute that box here, which I'm showing. Let's see if I can find the pen. A tiny edit machine score would compute this box, and then if you still exceed, if the scores here are not exceeding this box, then we are fine. So with this method, we ensured that we have 100% bit equivalent results with the software, BWMM software. And we could detect when we were successful, and in a very few cases where we were not successful, even with all these checks, with the edit score checks, we would throw that back to the host to run that alignment in the software. So that's how we managed to get 100% equivalent results. And then this helped to kind of improve the efficiency of a Bandit-Smith-Waterman accelerator by reducing the number of Ps we required and so on. And we had a working FPGA implementation of this on the cloud, so we could give it out and get it deployed today if we need to, because building a chip is expensive, but FPGAs, we can deploy our solutions there. So that was the read approximate string matching problem. But another thing which happens before the read alignment is finding so if this is the read, there could be two possible locations two or many possible locations, but in this example, there are two possible locations where this would approximately align. So instead of going and taking the string and aligning it across the whole genome, we've narrowed it down to a few spots using the seeding method, and then we can go and do the full expensive string matching for those locations. And what we found was this was very memory intensive. It's usually done using an index called FM index, which has a lot of random memory accesses, and the memory bandwidth requirements are huge. And that's where the bottleneck is. So so this is BWMM and BWMM2, and this is kind of telling the size of the index which we need to fetch, which is size of data which we need to fetch per read, and it's really a lot. So our observation was that can we build like a bigger data structure, but with more spatial locality, and so the more spatial locality and a better way of pruning off the searches through this data structure, so that we can reduce the amount of data which we need to move between the memory and the processor. So it was an interesting trade-off here where we are willing to make our index much larger. So I think the software folks, they went the opposite direction, right? Like, so this is a very interesting way where hardware software co-design can actually make a counterintuitive, different, take a different path, because all the research in read alignment was focused on making this smaller and smaller and smaller, this index, so that it can fit in the memory. And when architects look at this problem, we thought, okay, we have a little bit bigger memory capacity today, so why not expand this and build a more efficient data structure from scratch, and that would give you better results. And this, we built a specialized hardware accelerator for this, but the software itself was giving pretty good results as well. So ERT, we ended up, getting integrated with BWMM2, and so the software data structure itself can be used by anyone today. It's open source and is actually, there are many, we have seen some traction and many users for this. So the next step is sorting and marking duplicate, and this is just our, it's not intellectually very exciting, but then you have to make the step work and fast so that we can get a good end-to-end result. So we build a little a main memory sorting method and mark duplicates. It was just some software engineering here, but that we could reduce it from the current Picard tools to quite a bit once we hacked it out. The last step was variant calling, and variant calling is very similar to the DP problem for read alignment. Although the precision requirements and so there's some nuances and some differences here. What we found while analyzing this problem is that the entire DP matrix is like there's a lot of redundancy here, and you can prune a lot out. So the final optimal score can be obtained by executing much fewer cells, or much narrower, and it's not really a band, but much fewer cells. More than 90% can be pruned out while doing a per-HMM algorithm. So we developed a pruning algorithm and an accelerator architecture here too. And here as well, we had the threshold checks to make sure that we're not making any mistake, and we made sure it was again bit equivalent. And we taped out this chip as well and in 40 nanometer, and showed it worked on this specific algorithm and this specific problem, and had a software counterpart as well. So bit equivalence was a huge thing for us, so we had to do a lot of hang-ups and stuff to kind of make sure like everything was exactly identical to the software results. And this is a slide from Oliver actually, so the accuracy really matters, I think. I love this slide from Oliver, and bit equivalence is, we can build any number of accelerators, but if you want them to be usable and you want the bioinformatics community to use them, I think that should be an important guideline at least, right? So this is an example of how like a variant can actually make a difference. So if you make a many times there's like, okay, there's 1% accuracy loss, but how even like one variant which is different can make a huge difference is an example of that. So this is a CYP2C gene. It's used for metabolism, drug metabolism of blood thinners, Platviks. So here is a DNA which is normal, and on the right is aberrant. So this is the variant we are trying to identify. So there's a G to A variations here. So if you see this G has been flipped in this, this is our reference, which is normal, and this is the aberrant. Now let's see what happens if we make a mistake here, right? If there's an error and our aligner didn't catch it. So so what happens usually in prescription is that this GTAG pattern helps to kind of prune, cut down the non-coding region. So now we have cut it down, and we get the we get an RNA out of this DNA, and that translates to a protein which helps to metabolize the drug. On the right-hand side, if you see, because of this aberration, now we are going to splice a much longer region of the genome, and when the RNA is formed, it actually has a 40 base pair deletion, and therefore it is an aberrant protein which cannot metabolize this drug. So even the few percent or fraction of a percent difference in the errors can make a pretty big difference. So we should be careful about that when we do our accelerators. So finally, so what we found was by accelerating these kernels, which I showed you, we could get, this is on an FPGA system working on the cloud, so it's a real prototype. We could get it down to 2.5 hours, and because we kind of try to go end to end, and I'm not showing individual part speedups, but kind of showing the end-to-end speedups. So there's always Angel's law, right? This black was our Angel's law even, right? And even when we did ASIC, this thing was killing us. So and there's acceleration opportunities here, right? So we could build accelerators for these other stuff too. All right. So how am I doing on time? Okay, this is my second part. I'm gonna try and rush through that. I just have 5 minutes, I guess. But this is one project which we were very excited about, and it took us a lot of time to get through this. So, but let's talk about it a bit. And this we focused on long read technology. So, again, probably most of the people in the audience know about this, but we have moved from short read to long read now. And one key winner in long read technology is nanopore sequencing, where basically it breaks down the DNA strand, not breaks down, pulls the DNA strand through these motor proteins and then digitizes the analog signals to give us the base pair length. So here the read length can be even a million bases if you do the wet lab preparation well, but the accuracy is lower, so you need better error correction mechanisms. One thing which is cool about this technology is that it is portable. So you can kind of take it to point of care. You can build point of care solutions around it. So that's where we started. And the other thing which is cool about it is that it is a very nice filtration mechanism. So you can, let's say you see some part of your gene, of your DNA, and decide that this DNA is not relevant to your analysis. You can kick it back up. So you can eject that DNA back. So you can discard a lot of useless DNA which goes through it. So we wanted to leverage these two features, the read until and this point of care. So I'm going to talk about the point of care thing which we did in our lab, and then the kicking part, I would welcome you to read our paper, squiggle filter. So we built, what we realized very quickly while we were doing all this is that if we just focus on the computation side, we may not be able to, make a big difference. So we had to do a co-design of the wet lab sequencing as well as the software and the hardware. So we went ahead, got ourselves some minions, and set up a lab here, a wet lab in our in EECS, and this is Jack Warden. He was a postdoc in my group, and he really, it's amazing how he beautifully picked all this up, and then he he went on to become a postdoc in medical school. Now he loves to do this stuff. He doesn't want to do CS anymore, I think. But then, so the application, the point of care application, which was given to us by our physician friends, was that can we do intraoperative histology? So think about it like, let's say for brain, so for neurosurgery, when they're trying to remove malicious tumor tissue, what they need to do is they need to figure out the margins of this tissue, right? And brain is a very precious tissue, so you want to make sure you take just enough out, right? Otherwise, there could be a lot of cognitive issues for the patient. So today, how it is done is in a pretty primitive way. They take a slice of this tissue, look it up under the microscope, and then that's the feedback loop. So we thought, could we augment that, or could we replace that histology with sequencing? Point of care. But the challenge was to do this at about 30 minutes, right? So the whole thing from tissue to finding a hotspot mutation was to be under 30 minutes. But this was a very different problem from whole genome sequencing, where you have to find the variants across the whole genome. Here we know exactly what hotspots you're looking for. So one or two variants is what you're looking for. Fortunately, there's enough traction in the medical community to come up with a list of molecular parameters which can be used for this kind of diagnosis. And that was the first step. And then the question is, now can we do the tumor DNA in the interoperative time frame? So this is the pipeline. You take a piece of tissue, you have to do DNA extraction, and then you have to do some amplification of the gene you're looking at, do library preparation, sequencing, and then we do the informatics, right? So what we found was, when we looked at the time taken, this target amplification was actually huge. the PCR step, because and naturally so, because we're thermocycling here. We're changing the temperatures back and forth, which is a very mechanical thing to do, and so that's going to take up a bulk of your time. While if you can look at this is where we lived, right? This was really small. So first, a little bit of disappointment that the problem we are trying to solve is really small, while the bottleneck is somewhere else. So that's what motivated us to build up wet lab. But then what we realized is that we wanted to convert this chemical problem into a computational problem, and the way to do that is there are amplification techniques which are very fast but very error prone. So what we thought is, as informaticians and as like the hardware people, we can actually build error correction and the tools to kind of analyze that amplification product, make them very fast. So we can shrink this orange quite a bit, and that will make the computation time longer, but then we can accelerate that. So the way it went was that we figured out the first step was to do what's the bare minimum amount of amplification you need to do, so that we can do just enough sequencing. So what's the optimal point between the amplification and the sequencing time? First step was to find that balance, and that would lead to sort of an optimal time point. So that was the next step, and by doing that and also optimizing, it's amazing how Jack could even optimize the library preparation time. Like he looked at it and did some primer design studies to do that, and that came down to about 52 minutes. But still, in spite of this optimal thing, we still had a bottleneck there. So what we did was we used LAMP, and it's not a new technique, but it's not used in practice because of all these ugly little amplification sort of off targets and loops it makes when it amplifies the primers, right? So that was a downside. It was very difficult to analyze this, but hey, analysis is our strength, right? So we could tackle that problem. We can't change the thermocycling of PCR, but we can tackle this problem. So then we did with loop mediated amplification. So this is why it is fast is because it's isothermal, because you don't need to cycle between different temperature cycles to amplify the product. So, and then we developed like a tool called Lamprey, which would go and polish this, product and clean it up really nice and fast, so that we could actually get to the variants. We did this for one hotspot mutation, and so by doing both threshold sequencing and LAMP amplification, we could get it down to less than 30 minutes. So this is the final chart which we had. So we started here about 140 minutes, and then slowly with all the wet lab and sequencing and developing the LAMP protocol and the LAMP tool analysis, we brought it down to 30 minutes, which sets kind of the fastest time to result record for this specific diagnostic. So yeah, that sort of wraps up this point of care analysis, and we have released this genomics benchmark. So I just wanted to kind of throw this out there. If you're interested in this field and are looking for sort of wide set of computation kernels, which covered a lot of three pipelines. We cover pathogen detection, we cover whole genome sequencing, as well as long read polishing pipeline. So this is one resource which is available. We are planning to release a second version of it sometime soon after we wrap up the project I was talking about. But yeah, so this is not just my work, it's a collaborative work with a lot of very good people, including Satish, David, Jenna Weens, Robert, and Carl. So they have been our partners in the Prime, and they entertained us. They, like Bob used to make fun, like you are becoming more like me and I'm becoming more like you. So, but it was a fun experience, and an awesome group of fantastic students who were willing to take up this challenge and move fields and do what it takes to do good research. Thank you so much.
Great.
If you have any questions, happy to answer those.
Yeah, thanks a lot, Ritu, for this wonderful talk.
Sure.
Any questions?
I have one.
Okay, John has a question for you.
Sure.
The microphone to him.
Thanks. Hi. Hello, Ritu. It was a great
Hi.
talk and eye-opening, I would say, in many ways. So it was interesting that when you show the library preparation and the amplification time reduces significantly, but at the same time the sequencing time increases slightly, I guess. So this is probably because the type of application is making things harder, and then perhaps like the sequencing time is longer. So do you think, also based on your earlier work, the Skugel filter, do you think approaches like adaptive sampling, read until, these type of works will play an important role also to decrease that sequencing time, such that like we can also overlap the latency, right, while sequencing, to do the computation such that like maybe we can do the genome analysis within minutes. So how do you see basically the future of adaptive sampling in that scenario?
No, I think adaptive sampling is a very nice feature which nanopore enables, right, with the read until feature. So definitely it has a big role to play. So in pathogen detection especially, I think like when we are trying to get like a sample from host and then figure out like the, let's say, distribution of bacteria, or if a specific virus is present or not, there I think it will be very helpful, because a lot of the strands which would come through would belong to the host, and so if we can quickly identify that, we could filter them out. So that's very useful there. Now read until here in the project which I just showed you, like the cancer hotspot mutation. So why we didn't go for read until here, we thought about trying it out. But the reason was that we're targeting specific hotspot mutations, right? So the primers which Jack built were actually very short. They were not very long. So read until was not giving us a lot of benefit. Read until is useful if your sort of strands, the reads which are coming out are very long. So, very quickly you identify and then you eject it out. But in this case, our reads were short. And there was also, the reason to keep it short was, primer design complexity was getting very tricky. So if you make it very long, then you get a lot of false positives. And second, the library preparation time, that was very interesting. Like if you check our paper out, the library preparation time was actually lower for like little bit shorter reads, right? So that's the reason we didn't go for read until in the cancer, the real time margin detection project. Sure.
Okay. Any other questions for Ritu? Ritu, I think I have one question. First of all, I think it was a very nice talk. Wish you could be here in person, but hopefully next time.
Next time,
yeah, we could discuss more. That chart where you show the library preparation time versus all of the other stuff plus computation. How does that change based on the type of analysis you do or type of the genome? Basically, can you give us some insight into how these different portions behave based on the analysis type and genome type, etc.?
Yeah, no, that's a great question. So, so here, one thing what we are doing was, like, we are trying to, we did only one hotspot mutation, right? So we are looking at only one specific variant which we're trying to call out, and actually a SNP. So, that itself was quite challenging. But if we want to look at more like a broader panel, then the computation time is going to become significantly higher. And not only that, depending on the primer design, like which portions you're going to amplify, that is also going to become very tricky. So as you expand out the region of interest in the genome, then this, I can imagine that the computation time and the sequencing time will slowly start becoming bigger and bigger in this breakdown.
Okay, yeah, that makes a lot of sense.
Sure. And also LAMP is very error prone. Like, I mean, if you look at the kind of product it creates, it's very ugly. So, scaling that up would be a very interesting problem.
Yeah, certainly.
Cool.
Okay, if there are no questions, then let's thank Ritu one more time. Thank you, Ritu.
Of course, and it was a pleasure, and good luck with the panel tomorrow. Is the panel tomorrow in the?
No, it's on the 17th.
Oh, 117.
Yeah,
Okay.
slot time.
Yeah. All right.
Okay, thanks. See you. Okay, I think we have Hayu next. Okay, Hayu, are you
Yes.
there?
Yeah, yeah. Can you hear me?
Yes, we can see you and hear you. You can share your slides also. So our next talk is by Hayu Mao, who is a postdoctoral researcher at ETH Zurich in my group. And she's going to talk about the work she's presented in last year's Micro, which I mentioned today, this morning in the talk, in my talk, which is in-memory acceleration of genome analysis via tight integration of base calling and read mapping. Okay, Hayu, go ahead.
Yeah, cool. Thank you, Ona, for the introduction. And I'm Hayu. And today I'm happy to share our paper, GenePip, which is in-memory acceleration of genome analysis via tight integration of the base calling and read mapping. So in this talk, I will firstly give you an overview of this work, and then I go details about it. So for the genome analysis, it enables us to determine the order of DNA sequencing in the organism's genome, and it plays an important role in the personalized medicine, outbreak tracing, understanding the evolution, and so on. And modern genome sequencing machines extract smaller randomized fragments of the original DNA sequence, which is also known as reads. And in this work, we focus on the Oxford nanopore sequencing technologies, which is a widely used sequencing technology nowadays. And as you can see here, on the right side, it's like you can grab it with your hand. We focus it because it's portable. It provides portable sequencing devices, and it has high throughput, and also it's very cheap. We found that in the genome analysis in ONT, there are two main limitations nowadays. First of all, there are multiple steps in the genome analysis, which cause a large data movement between the multiple steps, and also it introduces a lot of wasted computation done on the data that is later discovered to be useless. So we propose GenePip. GenePip is a fast and energy-efficient in-memory acceleration system for the genome analysis pipeline via tight integration of genome analysis steps. GenePip has two key techniques. The first of all is the trunk-based pipeline, which provides a fine-grained correlation of genome analysis steps. And second is the early rejection, which timely stops the execution of useless data by predicting which reads will not be useful. GenePip performs state-of-the-art software and hardware solutions using CPU, GPU, and optimistic P. So I'll go to details about the genome analysis and our motivation. So this is the nanopore genome sequencing. After we using the genome sequencing machine, we get the raw signal genomes, and we store them in the storage. So we continue with the first step in the ONT genome analysis, which is called base calling. In the base calling, we trunk the raw signals into trunks and go through the deep neural networks and base call them into trunks, and then after that we finish in the read. Here we use the green one to represent the high quality of the base calling, and use the red one to represent the low quality of base calling. After we finish base calling all of the raw signal sequences, and we get all of the reads, and we store them in the storage. Then we continue with the next step, which is known as the read quality control. In the read quality control, if the read is a low quality read, then we discard it. And the read if the reads is a high quality reads, then we store in the storage and continue with the next step, which is read mapping. In the read mapping, we compare read to the reference genome and to see if it can be mapped to the reference genome or not. If it can be mapped, then if it cannot be mapped to the reference genome, then we discard it. If it can be mapped to the reference genome, then we store the mapping result to the storage. So let me go a little bit detail about the first the 3 steps that I mentioned before. So in the base calling, we usually use the deep neural networks to ensure the base calling accuracy. And the input of the base caller is raw signal trunks. And we translate the raw signal trunks to bases, which is known as the ACGT, and calculate each base quality at the same time. Then we assembled all of the base call trunks into a long read. So the output is a long read. For the read quality control, the input is the base quality scores of a read from the base calling step. And we calculate the average score of the long reads and compare with the threshold. If it's higher than the threshold, then it's a high quality reads, and if it's lower than the threshold, then it's a low quality reads. And the low quality reads are discarded. So the third step is read mapping. And in the read mapping, we first do the indexing of the reference genome, which is done by once and is done offline. So the input of the read mapping is high quality long reads that we generated from the read quality control. And firstly, we use the subsequencing reads to query the hash table to get the possible match locations. And then we identify the candidate regions and output the chaining score. And then we execute the final alignment step if there is a chain. So the output is the mapping information. Among all of these steps and operations inside steps, we observe 2 main limitations. The first one is the large data movement. We use the human data set as an example. So in this example, we observe that in the for the raw signals, we have nearly 4,000 gigabytes data, and after base calling, we have 546 gigabytes, and after the read quality control, we have 437 gigabytes, and after remapping, we still have 362 gigabytes. As you can see here, between each step, we have large data movement between genome analysis steps. The second limitation, we also use the same data set as an example to illustrate it. The second limitation is the waste computation. So after base calling, there are 100% of reads, let's say, and then after the read call control, however, there's only less than 80% are left. And after the read mapping, there's only less than 70% useful reads. So we conclude that a considerable amount of computation on useless data due to the low quality reads or unmapped reads. Let me talk a little bit about the state of art works. So the onion-based scheme is an efficient technique to reduce the data movement by processing data using onion memory. For the base calling, there's already some works to using the onion-based scheme, which is designed for the vector matrix multiplication operations, and which is a dominant operation in the neural network applications. Therefore, it can accelerate the base calling a lot. And for the read mapping, there's also work for using onion-based scheme for the search and addition operation, which is the dominant operation in the read mapping step. However, These PIM works reduce the data movement in a single genome analysis steps and exacerbate the data movement overhead between the analysis steps. And we conclude that non-prior work tackles data movement between analysis steps and reduces useless computations. So our goal is to efficiently accelerate the entire genome analysis step pipeline while minimizing the data movement and useless computation. And we perform a study to quantify the potential benefits, and the results are normalized to the performance of GPU. So we first consider the situation that we have NVM-based PIM accelerators for separate base calling and remapping steps. And it can choose 2.7 speed up. And second, we consider there's no data movement between these two accelerators, and it can achieve 6 times speed up. And third, we assume that there's no data movement and no useless reads in all of these accelerations, which is the ideal case, which can achieve 9 times speed up. So that's why we propose GenPip. GenPip is the first holistic in-memory accelerator for genome analysis pipeline, including base calling, read call control, and read mapping steps. GenPip has 2 key techniques: trunk-based pipeline and early rejection. First, let me talk a little bit about the trunk-based pipeline. Trunk-based pipeline enables fine-grained pipelining of genome analysis steps. It also processes reads at trunk granularity, which is a subsequence. Usually it's around 300 bases. Trunk-based pipeline increases the parallelism by overlapping the execution of different steps at a trunk granularity. Trunk-based pipeline reduces the intermediate data by computing on data as soon as the data is generated. Trunk-based pipeline also provides opportunities for the ER by analyzing a read at trunk granularity. So here we use an example that a read consists of 4 trunks. For the conventional pipeline, we base call all of the trunks one by one, and after base calling all of the trunks in this read, we assemble them, and then we do the quality control on the granularity of this read. And after that, we do the read mapping on this read. For our trunk-based pipeline, we base call the first trunk. Then when we base call the next trunk, at the same time, we do the quality control of the first trunk, and we do partial computation of the read mapping of the first trunk. So as you can see here, after we finish all of the computation of the trunks, and we do the rest of the computation of read mapping on the granularity of the read, and then we finish it. So you can see here, for our trunk-based pipeline, we can save a lot of computation cycles. So the second technique of GenPip is early rejection. Early rejection stops the execution on useless reads as early as possible by using a small number of trunks to predict the usefulness of a read. Early rejection predicts and eliminates the low quality and unmapped reads from the genome analysis pipeline as early as possible. So here I give an overview of the early rejection, how we do the early rejection. We base call small number of trunks and then check the granularity of these trunks, to check the average quality of these trunks. If it fails, then we stop analysis. If it passes, then we base call more trunks. And then we map the base call trunks so far, and then we check the mapping score, and if it fails, then we stop analysis. If it passes, then we continue base call the remaining trunks, and execute the remaining computation in the read mapping. So as you can see here, early rejection includes two sub-techniques. Early rejection based on the trunk quality scores, and early rejection based on the trunk mapping scores. So let me go a little bit deep about these two techniques. So the early rejection based on the trunk quality score, in this sub-technique, our goal is to accurately estimate the quality of the anti-read by checking a small number of the quality of a small number of sampled trunks. So here is the example of on the left side, the yellow one is low quality reads, and on the right side, the green one is a high quality read. And here, the red line is threshold 7, which is recommended by the statistics. So we find 3 observations. The first one is that the range of quality scores of the trunks extracted from the high quality reads is greatly higher than that from low quality reads. And the second one, single trunks quality score is not enough to predict the read quality score because there are many trunks whose quality score are larger than 7. The third observation, we found that consecutive trunks quality scores are usually close to each other, indicating that sampling consecutive trunks may not be representative enough to estimate the quality of anti-reads. That's why we conclude that we need to sample a small number of non-consecutive trunks evenly in a read to predict the read quality. For the trunk mapping, based on trunk mapping, the key insight of it is a read cannot be mapped to the reference genome if enough consecutive trunks in this read cannot be mapped to the reference genome. So if we map a small number of trunks, small trunks, it can provide too many possible mapping locations. So we found that to map a small number of consecutive trunks in a read, and then we merge the small consecutive trunks into a big trunk, and then we map this big trunk to the reference genome to predict whether the read can be mapped or not. It's worth to mention that the CP and ER can be applied on different systems such as CPU, GPU, and PIM. We implement CP and ER using PIM since PIM is more efficient to reduce the data movement between the genome analysis steps. We also apply CP and ER on CPU and GPU with science and observe the speed up and energy savings. So next I will introduce the GMP implementation. So GMP has 3 main modules: base calling module, read mapping module, and GMP controller. First of all, GMP controller received the raw signals from the sequencing machine and stored in the ERAM, and then send a single trunk to the in-memory base caller. And after that, we send the base quality score in the PIM CQS to calculate the quality score and of a trunk. Then we send the base quality trunk, the base code trunk along with the trunk quality score to the GMP controller. And after that, we send the trunk to the in-memory read mapping module, after we finish the read mapping, we send the mapping result to the storage. So when there is, we implement the early rejection by sending the quality scores to the early rejection controller. And if there is early rejection, then we send the base calling. Then we send the signal to the base calling module to end the analysis of the current read. And we also send the trunk mapping scores to the ER controller. If there's some ER based on the trunk mapping score, then we send the signal to both module and to exclusion of the carbohydrate. So these are the units that we designed, and you can find more details in our paper. And I want to mention one thing in the paper that we propose in memory seeding, because this can enable the very efficient seeding on different lenses. So basically, we after we receive the base code trunks from the GMP controller, we send it to the query string generator. Here we read a substring from a trunk, and then we use a shift register to generate the query string one by one, and send it to the RM-based CAM to search the key and find the match possible locations in the RM-based RAM. Then we send the possible locations back to the EDRAM buffer and forward these possible locations to the remapping controller. So it can efficiently support highly parallel seeding for long reads with variable lenses. And all of this, we managed to tightly integrate the genome analysis steps and to reduce the data movement and also eliminate the useless computation. So, okay, let's go with the evaluation. We used the simulation and also some simulators to estimate the performance area and power. And the baseline we use is the CPU, GPU, and the optimistic integration of two PIM accelerators. I want to mention here because we assume that for these two PIM accelerators, we don't have like the data movement. We assume that there's no power limit, there's no area limit for all of this. And the data sets are E. coli and human. Here are the results for the performance. So GMP can achieve 41 speed up compared to CPU and 84 times speed up compared to GPU and 1.4 speed up compared to the optimistic P. And we conclude that also in more details in the paper shows that both CP and ER are critical to the speed up. And for the energy efficiency, GMP provides 30 times percent over CPU, 25 times percent of GPU, and 1.37 times percent of optimistic P. And more details in the paper shows that ER is especially critical to the energy efficiency. And we also show that sensitivity analysis to show that how we choose the early rejection trunks and how many trunks we need in each data set. And this is designed when we analyze the one data set and offline. So as you can see here, for the early rejection based on trunk quality scores, we use 2 and 5 for the E. coli and human respectively, and we use 5 and 3 for the E. coli and the human datasets for the trunk mapping early rejection. There are more details in the paper, and okay, let's conclude our talk here. So the problem in this project we're targeting is the genome analysis pipeline has a large data movement between each genome analysis step, and there's also a significant amount of wasted computation on the useless data. So our goal is to tightly integrate genome analysis steps to reduce the data movement between the steps and eliminate the computation of useless data. That's why we propose GenPeep, the first in-memory genome analysis accelerator that tightly integrates the genome analysis steps. GenPeep has 2 key techniques, a trunk-based pipeline and a new early rejection technique. GMP part performs the state-of-the-art software and hardware solutions using CPU, GPU, and optimistic P. That's all for my talk, and I'm happy to take any questions. Thank you so much.
Okay, thank you very much. Any questions? No tough questions for Ayu?
I'm not taking longer than...
No, you did well, I think.
Okay, thank you.
Yeah. So I have one question. Why stop at read mapping?
It's actually a very good question. We can actually, remapping is kind of a necessary one if we want to know some information from the genome analysis. We have also the variant calling, but it's kind of not very necessary thing. We can also continue with the other steps, and there's actually a lot of things that we can do, and we focus on these three steps because it's kind of a very necessary step currently. And yeah, I agree with you. There are more things to explore for the other steps.
Okay, yeah, I guess that's good future work.
Yes.
Okay. Yeah, I think there are no more questions. So thank you for the talk.
Yeah, thank you.
Yeah, and I think that marks the end of our workshop today. We're going to have a special talk for John in the future for airlift. Like tomorrow morning.
Sure.
Okay, thanks everyone for attending. Hopefully we'll see you in the next incarnation next year.
Thank you, gentlemen, for a great workshop. That was really inspiring.
Thank you, Leonid. Thank you for staying with us. I didn't realize that you were
Oh, no,
here.
that was, I mean, I wasn't bored at all, trust me. I mean, that was very interesting.
Okay, yeah, thanks. And I will see you at ISCA.
Yeah, see you in June.
Yeah, take care.
Thanks again. Happy holidays. Bye.
Yeah, you too. Bye-bye.